Well, thank you very much. I certainly appreciate the introduction. Um, We're going to talk, I think, about body on a chip and the potential transform drug development. And I am both, associated with Hespros, and I'm emeritus at Cornell, but I still have an active research program at Cornell. And outlines a little bit of personal history. I'm going to emphasize physiologically relevant multi-organ systems, the interaction between organs that occurs. And a particularly important theme, I think, for all of us in MPS is the potential to measure human efficacy and toxicity simultaneously in preclinical trials. And I may speculate a little bit on the maturation of the field and possible paths forward. So a little bit about motivation. I started thinking about this back in 1989. I was on sabbatical at the University of Wisconsin. Most of my work at that time had been up into bioprocess engineering. My oldest daughter has Down syndrome. My wife wanted to cure Down syndrome, and I didn't think I could do that. But one thing we did recognize, and we've been told by physicians, sometimes they were reluctant to prescribe particular medications for people with Down syndrome because of the 21st, they had 3 copies of the 21st chromosome. And since the drugs had not been tested on them, it was unclear sometimes whether it was safe and efficacious, even though it had been approved by FDA. So one of the thoughts here would be, could we develop a system which allow a human surrogate to be used to test for drugs? And also, toxicology is very important too. And I'm not going to talk much about that here, but chemical toxicology, environmental toxicology, foods, cosmetics are all important areas potentially for the development of this technology. But I'm going to focus on preclinical drug development. And the question we're going to ask is, can we combine physiologically based pharmacokinetic models with in vitro mimics of organs to predict human response? And do this particularly, as I said, in preclinical trials. So the beginning was 1989. We had this concept. The first graduate student I had to work on the project, Lisa Sweeney, began in that spring. And we began working with John Babish over at Cornell's Veterinary College. And the first model we published on was this article in 1995. It was a macroscale model. We used little tubings and little flasks connected together, but basically to emulate the body based on a PBK. And I'm going to show a little bit more on that in a second. We did this for rodents, and we were interested in naphthalene toxicity. We did about 9 more papers using this macroscale system. But in 1998, Greg Baxter joined Cornell's nanofabrication facility. And he met with me the second day he was on campus, and we talked about taking this kind of system and going from the macroscale system to make it a microscale system. The advantages of the microscale system would be in terms of reduced cost and improved throughput, and that was the main motivation. The first description of such a device was in this little article that was in the Proceedings of SPE in 2001. But really, the 2004, these series of 3 papers were the ones I think that established this system and potentially its usefulness. It was a paper where we looked at, again, toxicity of naphthalene, liver, lung, and fat. And I might add that fat's an organ that's very important that is usually left out. Most of our studies leave it out too. But in terms of the dynamics of many of these drugs, which are lipophilic, is an extremely important component. With naphthalene, when we first started on the study, it was unknown what the reactive metabolite was that naphthalene formed, which was toxic. And why did mice, well, why were mice 10 times more sensitive to naphthalene than rats? So we used this system, and by the time we get done, and other people have found things in the time we were doing this, we were able to show why rats and mice differ, because we had the model and we had the PBPK. We showed that the difference really came from not differences in enzyme levels, but just in the presentation of drugs and the ability to regenerate the compounds, glutathione compounds, which would be protective for the lung, and the timing between rats and mice were different. And we are able to show again how fat modulated the presentation and affected the overall presentation. This led to recognition in Newsweek. They had a October issue which focused on what they thought were big new ideas. And it's one of the new ideas that they had. They're the ones who came up with the body on a chip term. We started building systems with both barrier tissues integrated with internal organs, 2009. 2009, it was a paper published from a Chinese group that used the term human on a chip. Now, up to this point, it may not be easy to recognize. There's a lot of resistance in toxicology and much more in pharmacology to any of those terms. So we usually use the term cell culture analog to start with. And that was less offensive to many of those people. But it was a human on chip was a term I was not brave enough to use back then. But once it was used, it was okay. I'm going to talk about our pumpless system. Up until 2010, all our work was with an external pump. The pumpless system has a lot of advantages I'm going to describe later on, and everything else we've done has been on the pumpless system. Also, 2010 was obviously a watershed year for the field as a whole. That's when Don Engberg's group came out with a paper, basically organ on a chip, and that had a significant impact on recognition for the field and funding for the field. And then Louise Marks' group had a paper that was the basis of the forming tissue use. He told me yesterday it was 2010 rather than 2012 it was formed, but the paper was 2010 on a chip-based multi-organoid system. In 2015, I had done a little work with Jay Hickman before this, but Jay and I agreed to activate Hespros to become a body on a chip company. And 2016 was really the first paper based on the work done with Hespros, taking kind of the multi-organ system we had, but combining it with Jay's expertise in terms of functional measurements, particularly electrical and mechanical forces that are being generated by the systems. Also in 2016, we did a paper on the side actually, demonstrating a 13-organ system. And the importance of that was not so much the system itself, but we could build a system for 7 days, 13 organs, physiologically, realistically scaled, and operate successfully. And we built the first blood-brain barrier model with authentic barrier properties. That's probably the more important paper then. The one problem with the pumpless system we had originally is that it was reciprocating flow. Flow goes down one direction, comes back the other direction. Not so good for endothelial cells. 2018, we published a paper showing how you use a pumpless system, but you get unidirectional flow through the device. The next two papers, well, three papers, really largely associated with my work with Hespros. A 2019 paper, Chris McAleer is sitting in the audience, and we'll be talking in the next session. Demonstrating that in human preclinical trials, it was possible to determine both drug efficacy and toxicity. And I think that's a theme I'll come back to again. The inclusion of functional innate immune system with circulating monocytes, we published in 2020. And what's important there, the pumpless system I'm going to talk about, is the fact that you can circulate cells without damaging cells. And I think that's an important point. In 2021, there was a paper published based on the Hespros system. I wasn't involved in this work directly, but the Hespros system was used by a small company called True North, which got out by another company which got bought out by Sanofi. But Sanofi took a drug that they were repurposing, using the data from the Hespros system for an IND, and began phase 2 clinical trials over a year ago on that. And I think that's the first case of using a microphysiological system as a basis for an IND. So the concept here that we began with is using mathematical models. Now, physiological-based pharmacokinetic models were developed in the late 60s, early 70s by Bob Dedrick, Ken Bischoff, using kind of a chemical engineering approach. divide the body into different organ compartments. You treat the organ compartments as either chemical reactors, absorbers, or holding tanks. And from that, you can predict the distribution of the drug throughout the system, time-dependent distribution of the drug throughout the system. And it was done largely originally for chemotherapeutic drugs to try to maximize the dose at the tissue of interest and minimize dose elsewhere, prevent side effects. You can also have pharmacodynamic models which says if the drug concentration is this at this organ, this is the response. And again, you do this not only for the parental compound, but for the metabolites. So we wanted to build a system guided by the PBK. And it's a little bit different than just being in a multi-organ system. It's a multi-organ system when it's guided by the PBK, where you get the compartments to communicate with each other in a realistic, time-dependent manner. And sometimes to build a totally realistic model is difficult, especially if you want to get 256 organs in it, which is not really possible. But what the PBK does of the device allows you to get the fundamental parameters that you can put into a model for a human or animal and predict what's going to happen in those systems. Okay, one other thing that's a little bit subtle here that I think sometimes is neglected is if you're going to model the whole body, you need to include all the tissues, at least all the fluid in the system, because otherwise you can't get the dynamics correct. In our case, the other tissue compartments off on a fluid reservoir, which you need to run the system anyway. But the other compartments is very important. And particularly for compounds where there's a fairly rapid exchange between the blood and interstitial fluid, the ratio of fluid is fairly large compared to the cell ratio. You can do this. It's much more difficult with the blood to cell ratio doing something which is realistic. But it's very important to have a low volume system so you can see the effects of metabolites on the other organs. So this is basically what we have. We have a system where we have the PBK, we have a physical system, we make predictions about what's going to happen. And some of the assumptions here is one, the organ volume ratios in the body are emulated in the device. More importantly, the fluid residence time per organ translates into fluid residence time per organ chamber. And why is that important? So you go back to chemical reaction engineering theory. It doesn't make any difference what size the reactor is. It could be 10,000 liters, it could be 100 microliters. As long as the residence time, which is the volume of the reactor divided by the flow rate into the reactor, you get the same reactants form, the same degree of conversion, same reactants form. Now, there's some subtleties in terms of absorption stuff on surfaces that you need to worry about as you change size. But in principle, that's our basis for scaling from a human to this device. Now, there's a lot more issues in scaling if you really want to go into it. And John Wixel over here, he could explain all the different possibilities. But for us, this made the most sense in terms of doing the scaling. We built a system where the fluid dynamics are well behaved. So we had passive fluid flow. We didn't need active valves. We could predict what's going to happen with the distribution of flow fairly easily. One challenge here is always the chip has to have cells which will grow in a common recirculating medium. And that is potentially a challenge, but we've always been able to find a way to do that. Sometimes it takes, you may mix three media, and I'll talk a little bit more about that later on. And again, this is where Jay Hickman's group also brings a lot of expertise, because a lot of it's not only the media, but how the media interacts with the surfaces, which is important. So we like serum-free media, and the company always uses serum-free media. In my lab, we sometimes use serum-containing medium. There's lots of advantages to serum-free medium, and particularly in terms of control over what's happening in the system. It's not that it's cheaper, it's actually more expensive to build a good serum-free media than using serum. But you again need to consider the whole system, and the surface chemistry is like I said, important. There's a couple of papers here which describe that. So what are the criteria to emulate a PBPK? The first four I've listed are relatively easy to achieve. I mentioned residence time as important in terms of scaling. Also, these systems have flow in them, and it's very important the flow rate past the cells be within a physiologic range. If it's if you static systems, many times the cells don't respond with the same physiology as they have in the body. They need some flow, but too much flow, of course, is detrimental. So it needs to be within the physiologic range. And that's one of the design parameters. The other thing, which is much more difficult to achieve, can be achieved, but much more difficult to achieve, is the ratio of liquid to cells and systems physiologic. You want a low volume system if you're going to look at the formation of metabolites and the action of metabolites in other organs. Now, there's a paper by from Mandy Esch's group at NIST in 2020 where she had a device that actually achieved a blood to cell ratio, which is physiologic. Most of the systems I'm showing you won't be fully physiologic that way, but they will be low volume systems so we can measure the effect of metabolites. The last point is the most difficult. Is the reason that these systems will never be perfect. And that's because once you take something out of the body, put it in a system like this, it's very hard to sustain the same physiology forever. But we're getting much, much better at getting tissue engineering constructs, which are much more mature. iPS cells, I think, are going to be ideal. I'll mention that at the very end. So there's been improvements that way. But again, the device need not be perfect to be useful. If I look at drug development, for many compounds, you're talking about 10, 11% of the drugs that are tested going into preclinical trial come out as approved useful products that are marketed. And if we can change that ratio from 11% to 33%, that has a tremendous impact economically. It now costs you presumably 1/3 less to develop the drugs. You're not spending clinical trials on something that's not going to work. You know that ahead of time. So society gains in potentially getting many more drugs and many more drugs at a cheaper cost. And that's one of the real potential benefits of this. So even though it may not be fully perfect, it can provide guidance, which is, I think, very important. Now, I'm not going to spend much time on this. Don Einberg had a nice paper on this, but basically, you can have engineered systems where you put a membrane down, you get the cells, you put them in the right positions, etc. You have organized, which are basically self-developing. Both can be useful. We generally use mostly engineered systems because we have much better control over what's happening. We do the measurements much more easily. We do use organoids in some cases. And Don had this paper in 2000, I think it was 18, where he talked about some of the advantages of engineered systems versus organoid systems. They both have advantages and disadvantages. You need to be aware of what you want to do in terms of what you might choose. One of the things that's important, and again, that you have to always do, and the company always does, is run the system for 24 hours without the cells to understand your drug losses due to absorption and adsorption. And from that, then you make some prediction about what's going to happen. What are the doses you really need to have the dosage in the recirculating fluid? And it doesn't mean solubility because you can have proteins in there and they'll absorb to the proteins, etc. But what's going to be the concentration in recirculating fluid is going to be presented to the cells. And again, for some compounds, you don't really have much losses. For some lipophilic compounds, you can have significant losses. So again, you need to do this kind of experiment to anticipate what's going to happen. Again, this is a case where on the left-hand side, the model predicted from that data what we thought should be the concentration. And then when you compare it to the measure values, they come out basically the same. So in 2010, we published a paper on the pumpless system. And it's not only pumpless, it's self-contained. And sometimes people talk about having a little chip and then they have a whole bench full of equipment to support it. This is a device that can act by itself. It don't have to take it apart, etc. It can operate for 28 days by itself. So the basic idea of the pumpless system is you have the system and you have two reservoirs up here and here. And the reservoirs act against the other tissues compartment. You tilt this flow by gravity down here, driven by this degree at which you tilt. And then after you do that, you flip it, it comes back the other direction. So you do get reciprocating flow, and we'll talk about consequences there. But for most cell systems, particularly internal organ, reciprocating flow is acceptable. You can predict the flow rate very easily. The solid line is the Higgins-Purcell equation. The dots are our measurements of the flow rate. So over a significant factor of heights here, we can predict the flow rate through the system fairly precisely. The gravity-induced flow is very important. One of the biggest problems we had originally, operating systems for a long period of time, was the formation of gas bubbles, and the gas bubbles plugging up the microchannels. The gravity-induced flow gets rid of most of this bubble problem. Also, it's much more efficient to assemble and operate. Now, one thing I want to point out, and just as in general for all these devices, is if I ask you to go 28 days is our goal here because that matches what happens in a lot of drug development. If I ask you to go 28 days without eating, drinking, or going to the bathroom, you would have problems. Okay, this device would have problems too. So we try to emulate that in a crude way by removing a portion of the fluid from the other tissues chamber each day and replacing it with fresh fluid. The fresh fluid will replace the nutrients that have been consumed, remove some of the waste products. And we do that for all these systems, and we can operate them for 28 days or longer in this kind of system. So there's a lot of advantages here. Again, we got rid of the external pumps, but allows continuous recirculation, eliminates the tubing. Before, if we had external pump, we often had tubing. If we have tubing, that's a source for non-specific absorption. You want to reduce the non-specific absorption as much as possible. It's a low volume system, and it's particularly you get rid of extra volume by getting rid of the pump a lot of times. That means you have a system that allows you to evaluate both the parent and the metabolites. And that's very important because most of the compounds we're interested in looking at is not the parental compound, but it's the metabolites formed by the parental compound, which are important. The main reason we did this, it was robust, you can operate for a long time, 28 days, again, no gas bubbles, no moving parts to fail, so you have a high success rate. We tried doing this with a pump system to operate for 28 days, you lose almost all the systems before you got to 28 days. It's self-contained. Again, once you put the system together, you remove some sample from the reservoir, but that's about it. You remove sample, the sample you remove to emulate the waste removal and feeding, that sample becomes something you put in HPLC and mass spec. Okay. You don't have to take it apart because we're also going to talk about these functional measurements and their electrical and optical measurement. You don't have to take the system apart to measure it. You run many units in parallel. A rocker platform is pretty cheap. You can run, run 9 units on a platform. You run 4 platforms on a rocker if you want. You can run a lot of units on such a system. The other thing which is important is that many times you want to look at different cells which circulate in the system. If you have an external pump in the system, almost always that pump will damage the cells or cause them to be activated when you don't want them activated. This facilitates recirculation primarily of immune cells and circulating tumor cells, the ones we've been interested in. I'm not going to show any data today on circulating tumor cells. It's something we're doing right now, but those experiments are ongoing. And the last thing, Jay Sung was the student working on this, and he had a vested interest in doing this because this decreased his time in the lab considerably. He put together 24 systems much more quickly than he could before. In about 1/8 of the time before, you can get all these systems together than they could with the pump system. So the other things we've been doing to make the pumpless system work better, self-contained system. One is constructs which are more and more authentic. We do the integration of barrier tissues, and I mentioned the unidirectional flow, which we'll talk about a little bit. Again, the serum-free media control surface chemistry is very important in this. Then I might add, again, that this is a modular system. Some tissues may take 2 days growing and to maturity, you can put them in the system. Others may require 21 days to grow to the point of maturity, you can put them in the system. You grow them to maturity and put them together in the system when they're mature. It also makes it a little easier to find a medium because you're not trying to grow the cells any longer. You're trying to maintain the mature metabolically active phenotype. So how do we monitor these systems? Well, we look at transepithelial electrical resistance for the barrier tissues, and we can do that in a sustained way. All the devices we make have optical access. So for example, if you have a fluorescence compound, you can look at the distribution of fluorescence compound in the system, et cetera. But we do the sample from the reservoirs, like I said, and that provides enough material to go do HPLC mass spec on, get a chemical measurement, not only the parental compound, but metabolites or biomarkers that we need. But again, the functional readouts, I think, are even more important. And I'll talk about that, but this is really what Jay's group has contributed. The electrical activity and force generation of neurons and cardiac tissue is particularly important. Neuromuscular junctions is one thing we do a lot of work with. The functional readouts are non-invasive. It facilitates chronic monitoring. Again, if I'm going to operate for 28 days, I'm going to be looking at responses that are, the acute response requires a certain dose. The chronic responses, you usually have less dose, will give you a certain effect over a longer period of time. If I'm going to emulate what's happened, if you're going to take a drug every second day, I can do that with this kind of system. And it's a rapid, direct measurement of response. Sometimes, if you rely on cell viability, many times cell viability doesn't change, but the action of the cells change. In terms of response, you can see responses in terms of minutes, in terms of electrical activity and force generation many times. And it can be diagnostic, it can lead to a better understanding of underlying mechanisms also. So this is just an example. And what I want to emphasize here is often people talk about 2D versus 3D when talking about microphysiological systems. I think it's much better to talk about organized and disorganized cultures. This is an example of a 2D organized culture. Now, if I take the cardiac cells and just plate them, I can't measure conduction velocity or anything about the electrical activity because it just gets dispersed. But if you control the surface chemistry, I lay it down like this U-tube shaped thing that's here. You can force the cells down here. And if you make this narrow enough, then the electrical activity is forced to follow this line. And that leads to the ability to measure things like conduction velocity, QT interval in the heart, etc. So although this is a 2D system, it's an organized 2D system and gives you real functional information. Again, this shows this. Again, I can stimulate here with electrical response, and I can just follow what's going to happen in the system. And it's you can, the change in conduction velocity in response to the drug can tell you a lot about the action of the drug. Or a chemical if you're looking at toxicity. Measuring force, you can use these cantilevers. And the way we do it now is largely a cantilever. The cells are going in the cantilever. They contract, cantilever bends. We measure the bending using basically a laser. And we look at the deflection of the cantilever. And from that, you do the back calculation to the forces generated by the cells. This is a little awkward sometimes to do on large scale. So we've been moving towards a piezoresistive method. I don't have any papers yet to describe this in any detail, but this is what we're working on right now. The piezoresistive method would be even easier to implement than the one with the laser, because you have to move the laser around, ratchet around to see all the cantilevers, etc. Well, so this was first system, again, I think basically Hesperos put out. It was a 4-organ system. It had a liver compartment. Liver compartment can be 3D. You're looking at making your metabolites. And again, you have a reservoir here and a reservoir here, and they're going back and forth. Now, the neuronal part, the skeletal muscle part, the cardiac right here. The cardiac right here is electrical and mechanical. And you're measuring electrical activity and mechanical activity here. We're not measuring, we don't have the 3D cultures there. We have 2D cultures there. We're using these to measure the response of the body to particular metabolites. We're not looking at the generation of metabolites because you would need the full 3D structure to do that. But this is diagnostic in terms of the response. The liver is the one thing that we do have to worry about, the formation of metabolites, recirculation. Again, this is where if you take samples out of the thing, you put an HPLC, mass spec, you can measure the overall response. Okay. Now, one thing again is 28 days is a fairly standard operation for extended studies in preclinical trials, mostly with animals. We wanted to show that the system could operate 28 days and maintain a fairly stable baseline. And that's what this described in this paper. And because we have a stable baseline, we can look at the difference between acute and chronic responses to different drugs or chemicals or whatever you wish. So both efficacy and toxicity are important. This is a paper that Chris McAleer was the first author on. It was between Hespros and Roche. And basically it was to evaluate the human cell-based in vitro system, both for on-target efficacy and off-target toxicity of parental compounds and their metabolite. And when done with 3 drugs, as an example, we'll look at tamoxifen and verapamil. And this is basic system. It was cancer, and we were literally looking at 2 different kinds of cancers: multi-drug resistant cancer and sensitive cancer cell line. And the liver and liver system, because we're looking at the formation of metabolites, that's important. And the cardiac was potentially in terms of side effects. In terms of drug side effects, and many that are important, cardiac and liver are 2 systems where drug side effects are really often very critical. This just shows tamoxifen, an anti-cancer chemotherapeutic agent. If we look at the sensitive cell line up here, you see there's a gradual, tamoxifen by itself is not toxic, is tamoxifen metabolites are toxic to the cells, kill the cells. 4-hydroxytamoxifen right here is the important one. You can see right here, for the sensitive cell line, as you increase, we see increased activity. With the drug-resistant cell line, this is a drug-resistant cell line because it overproduces P-glycoprotein. It's a pump protein. It prevents the input of the chemotherapeutic. If you have verapamil, verapamil suppresses the pump protein, allows the drug to enter, and then you see a big response to the drug. So you see the difference that way. You start to get a sense of the efficacy of the drug here. Also, you can look at side effects. These are cardiac side effects here. You're looking at beat frequency, conduction velocity, force generation right here. And again, the idea here is that there's very little change in viability. There's significant changes in terms of the functional response. And again, basically, if I look at the liver in the system, in terms of urea and albumin production, it's fairly constant. There's not any sign of toxicity there. So the toxicity is specific towards the cancer. Okay. So can we ask the question, can we use the MPS to predict PK/PD relationships? This is done jointly with AstraZeneca. I'm not going to go into much detail on this one. But the point here was, this was a drug which was confounding for them because the maximum drug effect was not driven by the peak drug concentration. There was a time factor. Drug was added, it was absorbed, then they had an effect. So there was a time can be a critical factor in that. That's often not uncovered until you do advanced preclinical trials. You'd like to know that ahead of time. This is the kind of system that can allow you to do that ahead of time. And we did those studies with turfidine. We did it with multiple species and showed that we can extrapolate between species. Fexofenadine, turfidine was withdrawn from the market because of cardiac side effects. Fexofenadine, you know, is Allegra, and that's the product of interest. And you can show, for example, there's 2 orders, you can't really see it. There's 2 orders of magnitude difference in the response here. Just the cardiac system there's significant response. Once you put the liver in here, it metabolizes terfidine to affects the phenidine and relieves the inhibition. So but if you're a person that has liver activity which is less active, then you have heart side effects. And again, one other thing that was in this paper, which I think is important, is that AstraZeneca had a compound, and they assessed it by their normal studies, not with flow or anything, quiescent studies. And they thought it was safe in terms of not having in vivo QT effects, up to 100 micromolar. But then again, the run in animal testing, particularly higher animals, they found that the QT interval increased by 22%, only 3 micromolar, which is unacceptable. They didn't discover that until fairly advanced preclinical studies. And the reason for that was it was forming a metabolite. But again, we showed that if they used the Hespro system, they would have found that 3 micromolar was in fact toxic ahead of time. So again, shows potentially the value using the system ahead of time. Another thing about this one paper that I didn't really emphasize is doing lots of extrapolations. They were able to convert the data from the device into PK/PD relationships and make mathematical predictions. This is another paper, again, done largely by the group at Hespros, but done in conjunction with Hoffman LaRoche, looking at recirculatory monocytes. And again, we wanted to look at tissue-specific damage and holistic pro-inflammatory response. But what was really key to making this system work was that you can maintain the system for 7 days, recirculate the monocytes, and you could do that without activation due to flow. Most of the time, first of all, a lot of the systems which would have pumps would kill the cells that were circulating. But even if you had a system which was gentle enough not to do that, often that movement would be enough that would kill the cells. Using a pumpless system allows us to do the recirculation in a way which is sufficiently general that we did not activate the monocytes due to flow. We did when we added chemicals, et cetera, to the system. And basically what we could do is show that we get the M2 phenotype and the M1 phenotype in the system. Again, we worked on some other recirculating cells. But again, I think the pumpless system, this design, is very helpful in working with cells when you're worried about inadvertent effects due to flow. Okay. Now the barrier tissue organ work, and this is barrier tissue. So we also have a lung model I'm going to mention at the end. This is mostly done at Cornell. The last 3 things I talked about done largely at Hespros. But Hespros has adopted a number of these things. Now we've done actually a lot of work on the GI tract. I'm not going to show you any data from the GI tract today. I'm going to concentrate on the blood-brain barrier. We worked with Sean Polachek and Eric Schuster at the University of Wisconsin, and they had developed a way to take iPS cells and derive brain microvascular endothelial cells. And they had built a preliminary model with of the blood-brain barrier using the brain microvascular endothelial cells co-cultured with astrocytes on a porous membrane. We took that system and we built a blood-brain barrier. Now we had authentic residence time, same residence time as in your brain was in this model. What we have here is, and you probably can't see it because the slides aren't going to be large enough, but the astrocytes on one side, membrane in the middle, and the brain microendothelial cells on the bottom. Now you also notice this is indented from the flow. It's indented from the flow because the fluid effects can be significant. We put it back so the shear forces on the brain microendothelial cells were within the physiologic range. And if we did that and operated the system, we have, I think for the first time when we did this, authentic values, the sustained authentic values of the TER in the system. The TER for the human blood-brain barrier is somewhere between 1,500 and 8,000 ohm centimeters squared. And you see that we're up to 4,000. We're sustained around 3,000 for 10 days, a little bit longer. And you can see that the tight junctions are also measured here. So that we have a system which we think very effective in terms of emulating what's in the blood-brain barrier. You can also look at drug uptake to the blood-brain barrier model. And we're looking at 5 orders of magnitude here, difference with these compounds. And you see that there's a pretty good correlation between the log P, the permeability basically in the chip and in vivo. That this is, ideally be a 45-degree line. So we think we can build a blood-brain barrier, which is actually fairly authentic. Now, one thing I'm not going to go into because I don't want to take the time to do it. If you want to, you can look at this one paper. But basically, the rocket platform, before the flow goes down, flow goes back, and reciprocating flow. So we still have to have that flow like that. But we can rearrange the system. And again, Yi Wang was a very clever postdoc with me. Oh, sorry. Basically, this middle channel right there, you can have flow always the same direction if you do this right. And part of that's based on here, and I can't shine the pointer through the marker there, but basically there's those 2 circles there. There's 2 outlets and inlets in each of the chambers. When you flip it this way, the fluid goes on down, but there's enough fluid retained by capillary action. And then it acts as a passive valve, closes the valve. And by doing that, you can change direction of flow here. So you always get the flow going one way through the system. And again, that has a significant effect physiologically. This is unidirectional flow. You see the cells align, direction of flow, and then diffuse cells. Now, this is a model where we just had UVEC cells in the channel. I'm going to show you one result where we have multiple organs in there. The bidirectional flow, you don't see the orientation. You can see this measuring angular orientation, quite a bit different. It also has an effect in terms of the physiology of the cells. Now, when we put these systems together, we don't necessarily want them to grow any longer. In this case, you see we put them in here, and with the unidirectional flow, there's no more, the growth is minimized here. You get sustained growth in the system with bidirectional flow, and even a little bit more growth when it's in a static system. So again, you can obtain a more physiological system by giving it the appropriate forces. We did this with a 3-organ system. That was just with the vascular. We did a 3-organ system looking at response to Tegophour and Uracil, which is a cancer treatment, colon cancer, bone marrow and liver. And basically everything we saw in this system was consistent with the known response of to that drug. We've also built a breathable lung. Now, Don Engberg presented a breathable young lung yesterday, and did it many, many years before this. The difference here is this is in the context of a multi-organ system operated at the same time. But again, operating so you maintain an air-liquid interface moving back and forth takes some engineering design to get that to work right. We did this with lung, liver, and breast cancer. We looked at the treatment with one chemotherapeutic. The question we asked here is, would inhalation therapy be as effective or more effective than intravenous therapy? Potentially, inhalation therapy could be done as a home-based therapy. The intravenous therapy pretty much has to be done in a hospital setting. So it'd be a big advantage potentially to doing it with inhalation therapy. And what you can show is that the system is, in fact, we can do recirculation. Again, if static treatment, you won't see any of this. You need also the movement that Don talked about in terms of seeing the appropriate response. But we did find that intravenous therapy resulted in a much greater killing of the cancer cells, 49% death versus 21% death for intravenous, and liver and lung death was insignificant. Okay. Just mentioning again, increased complexity is possible. Again, this was just to show that you can put the system together, 13 organs, 3 external barriers, 2 internal barriers, 8 internal organs, everything's organized proportionally, except fat and muscle. And again, they were 1/12 their normal size to fit onto the chip. But otherwise, we could make this sustain for at least 7 days, etc. This again reminds me, we left fat out of most of the chips I talked about. But fat can be really important in terms of overall dynamics here. So adding fat back in would be a good thing many times to do, particularly for any compound which is lipophilic. We can apply this to rare diseases to get work done with Virginia Smith at Hespros, applying it to a rare disease. And again, we think rare diseases is a opportunity for MPS systems to establish a toehold in terms of their ability to provide useful data in a regulatory setting. Myasthenia gravis is a chronic neuromuscular disease. Autoantibodies block this receptor. We look at a neuromuscular junction. Jay showed this slide yesterday, but basically, or something similar to this slide. But right here, you have the axon bodies, or the neuronal cells here. They form axons, and they have tiny channels. The cell can't go through, but the axons can penetrate. The axons penetrate to the muscle cells over here, innervate the muscle cells over here. So you get a neuromuscular junction system where this can respond to things on this side or this side, etc. You look at drug response. If we do this with this system as a model for this disease, we see the treatment with the antibody results in 3 different kinds of responses, all which are observed clinically. And so it does this in a way which can be used to test potential treatments, etc. Now, in terms of supporting clinical trials, this I was not involved with. This is a paper, and actually, Chris McAleer is going to talk, I think, later this morning about the system, this paper, basically. But the data from this paper was used to provide efficacy data to gain regulatory approval again for this clinical trial for Sanofi for a repurposed drug. And the phase 2 clinical trials were for these 2 neuropathological diseases. But I think this is, again, the first example using MPS to gain regulatory approval. And I think that's an important step for the field. And again, Don Engberg talked yesterday about applying some of this to COVID-19, which would be another example of using MPS systems in terms of regulatory approval. And so I think drug efficacy can mean a lot in terms of regulatory evaluations. And again, you can start thinking about doing things for with MPS systems for groups which are not easy to do in terms of clinical trials of children, aged individuals. Ultimately, ultimate precision medicine is potential with these systems. You can design your clinical trials to target certain subpopulations, etc. In terms of further development, the things I show are things that people are doing right now. It's not that they're not doing it, but these are areas in which I think there's more and more emphasis on. There's going to be more and more sophisticated models being developed for the immune system, circulating tumor cells, reproductive tissue. The neurological disease is important. It's one that we're very interested in, but I think there's going to be, again, greater maturity in terms of its application in that area. More authentic tissue constructs is certainly something many people are working on. They can be incorporated. I'm going to talk a little bit more about IPS technology, but I think that's going to be an important technology for this. And again, personalized medicine, precision medicine. is a way that we can ultimately go. I don't think we're really there yet, except for very specialized cases possibly. So iPS cells, I think iPS cells are particularly useful for MPS models. And that's because now you have a controlled cell source that replicates many of the advantages of primary cells, but no, without the variability. So now I can run, hundreds of experiments with basically the same cell base. And that's probably important in terms of really understanding what's happening in the system. I can do this to represent different genotypes. I get iPS cells from different populations. I could go back to the idea of Down syndrome in that. So we can mimic disease tissue as well as normal tissues in these models. And again, personalized systems. I won't spend much time on that. I think that's fairly obvious on that. And so in conclusions, basically, I think we can build physiologically relevant multi-organ systems with continuous recirculation, serum-free medium. And that's what we've shown, and I think that's desirable. The functional readouts, I think, are important because it gives you a way of looking at what's happening very rapidly to application of drugs or chemicals. If I'm looking at chemical toxicity, it's very important that way also. It can be diagnostic. It can lead to even better understanding. In the heart, does the drug affect the electrical activity, the force generation, or both? I can start getting some insight that way. The pulmonary systems, I think, are very important. The barrier tissues, I think, can easily be incorporated with internal organs. And then I think for the field as a whole, looking for a toehold in the regulatory environment, I think looking at diseases, probably particularly rare diseases where there are not good animal models, and using drugs which already have been approved for human use, but not necessarily active in human use, looking at using the system for efficacy in those kinds of systems is actually a good model in terms of gaining a toehold in the regulatory environment, gaining confidence from the pharmaceutical industry that this can work. Also, in terms of things like foods, cosmetics, this also would apply as well. So again, this is support, and I just want to thank all that. Again, I might mention one thing about support, though. The first 20 years of my work in this, all the support had to come from NSF and the Army, because it would never get past NIH review at that point. It was really only after about 2010 that there was a reception at NIH for this kind of studies. We did submit to NIH, and you can get 3 good reviews and 1 bad review saying that it would never work, basically. So all our early work was supported by NSF and the Army. So with that, I'd like to, I can take questions if there's time. If not, I appreciate your attention. So thank you.